Bismillah ar-Rahman ar-Rahim. We'll start with the first chapter on paper 4, which is the A2 syllabus. And the A2 syllabus, the first chapter is respiration. And we've got videos on respiration, but then now I'm going to discuss the questions on respiration only, as asked by many of you who leave your comments at the end of my YouTube videos. So I'm starting with the questions on respiration. First, you need to revise the chapter on respiration and then start doing these questions. Now, if you look at the first question that we have here. Now, the first question is ATP is used or produced. Now, if you look at question number 1, ATP is used or produced at different stages in the respiration of glucose in aerobic conditions. Complete the table to show whether ATP is used or produced at each stage of respiration. Write yes or no in each box. Glycolysis, yes, ATP is used and it's also produced. In link reaction, no ATP is used, but it is not even produced. Krebs cycle, no ATP is used and it is produced because we have substrate level phosphorylation here. Here also we have substrate level phosphorylation. Substrate level phosphorylation here, substrate level phosphorylation here, substrate level phosphorylation. Then oxidative phosphorylation, no ATP is used, but ATP is produced because all the NADH move to the inner membrane, and there, of course, the ATP is made. So this was a very direct question, and I'm sure everybody would have got the 2 marks for this because the 2 columns, you see, you had to get 2 marks for the 2 columns that you would get the 2 marks. So 4 pairs, 2 marks, 2 or 2 rows correct, 1 mark. If ticks and crosses used, then I need all the 4 correct for a maximum of 1 mark. So there were 8 points to be correct and you had to get 2 marks, so 4 would give you 1 mark. Okay, now as you look at the later part of the question, now please remember paper 4 questions are to be read very carefully and very slowly and have to be read at least twice. An experiment was carried out to investigate the effect of epicatechin or whatever it is on mitochondrial respiration in mice. Epicatechin is naturally occurring compound in cocoa beans and so is present in chocolate. 2 groups of mice. So we had one group of mice A, and then we have another group of mice which is called B, were used in this experiment. 2 groups of mice, group A and group B, were used in the experiment. Group A was given water containing epicatechin. So these were given water which contained a chemical epicatechin twice a day for 15 days. 15 days, subah sham. Subah sham means morning and evening. And group B was given just water. Here it was water plus epicatechin. Here it was water but no epicatechin. Twice a day for 15 days. So they were also given subah sham and they were also given for 15 days. So the common thing is twice a day for 15 days and twice a day for 15 days. Both had water. In one the chemical was added and in one the chemical was not added. So you have to spend time in understanding the question. After 15 days, the structure of mitochondria from striated muscle cells in both groups of mice was examined. The surface area of the inner membrane of the mitochondria was divided by the surface area of the outer membrane to obtain a ratio for each mouse. Now, this is a mitochondria, and then this is the inner membrane. Now, the surface area of the inner membrane and the outer membrane is being compared. Inner membrane was divided. So inner membrane was the one which was the larger number because it was folded. So say it was 10 surface area and the outer say was 5. So 10 ratio 5 means 2 ratio 1. Always give yourself an example to explain this to yourself. So 10 ratio 5 would mean 2 ratio 1. So table 4.1 shows the mean ratios for the 2 groups of mice. In A, it was the inner membrane was 20 times the outer membrane. No, sorry, 2.0 times. So it was 2.0 times to 1, and this was 1.7 times to 1. So in A, mice A, where the chemical was given, the inner membrane was slightly more, had more surface areas compared to this. While in the B one, in which just water was given and no chemical was given, they had 1.7 ratio 1. The mice in group A the mice in group A were able to exercise longer than the mice in group B, right? Now, all this information is very, very tough information because we haven't studied this before. So, you need to figure this out. With the reference to table 4.1, explain why the mice in group A were able to exercise for longer than the mice in group B. So, I mean, just a quick revision. This is the outer membrane. This is the inner membrane. The inner membrane is thrown into folds, which is called the cristae, and inside the shorba or the fluid is called the matrix. And then you also have a loop of DNA here, and you also have 70S ribosome, right? So if the now what is present on the inner membrane? This is what you need to understand. What is present on the inner membrane is those channel proteins. There are three. That's called a multi-enzyme complex. And then you have the famous ATP synthase. So the ATP synthase is present on this, and so that is what is going to result in the formation of ATP. So please understand the question with the explain why group A were able to exercise for a longer time. So table 4.1 was this, that A has the inner membrane has a larger surface area. So the inner membrane has a larger surface area. So the inner membrane is 2.0 times, and this is say 1 time. So say this is 20 millimeter, no, that is 10 millimeter square surface area. So give yourself an example and try to understand the question. Now, if you use the word they, now what does this they mean or the means? In the question, as group A is coming first, so we will assume as examiners, if I'm the examiner of this, I'll say they means group A. So whenever they are comparing two groups like group A and group B, please specify which group are you talking about. Are you talking about group A or group? If you say the or they, then we are going to assume you're talking about the first one. So let's now, I always do that is, I always ask you all to write down whichever group you're talking about. So just write down group A and then, just put a dot and then put something like this. So group A has a higher ratio. And this is now you're referring to table 4.1. So it has a higher ratio, 2:1. Now just writing that got you 1 mark. So this is the first mark scheme point. Then the second mark scheme point, it says larger inner membrane. You see, as an examiner now, when I'm telling you the mark schemes, I know when I'm checking papers, nobody writes it in this order. Everybody will have written whatever comes to their mind. So the inner membrane has a larger or a more inner membrane or more cristae, larger inner membrane than B. Well, look at B. This was 2.0:1, and this was 1.7:1 because you are referring to the table 4.1. So that gets you another mark. Larger membrane slash cristae than B, then more ATP synthase. Why? Because there will be more ATP synthesis. There will be more ATP synthase, or you could have said cytochromes, or you could have said more electron transport chains, carriers. or you could have used the word stalked particle. So if you're familiar with these words, I'm fine with that. You can use these words. I'm not saying it's not allowed. Then the next. Now this will be, what is going to take place here is oxidative phosphorylation. So there's going to be more, there is going to be oxidative phosphorylation taking place here. And what is that going to result in? That is going to result in more ATP being produced. So more ATP being produced. You don't say more energy produced. Energy cannot be created or destroyed. So more ATP produced. Muscles can contract for longer. Muscles can contract for more time. ठीक है? And then you could have said anything else like, hydrogen ions move down their gradient. Any little bit of explanation about that. So you see how many points there were. There was one, 2, 3, 4, 5, 6, 7. Now you only have to give me 5 points out of these 7. So any 5 out of these 7 would have got you your 5 out of 5. So remember, you always write more so that you are very clear that you get your full marks in this sort of a question. It's a very important technique which you must follow. Coming to question number 2, figure 1.1 represents the link reaction. And it's R plus coenzyme A gives you NAD is converted to reduced NAD or NADH, and it forms S plus acetyl coenzyme A. Now name substances R and S. R was of course pyruvate and S was carbon dioxide. Now this was a little technically difficult. I agree with you if you say this was difficult. So R is pyruvate and S is carbon dioxide. Decarboxylation takes place here and we need an enzyme decarboxylase. Then it says explain what happens to the reduced NAD. Now this reduced NAD, what happens to this? Where does it go? So explain what happens. When it says explain, well, you need to give me a biological explanation. Explain, and that's 2 marks. So always think of the 2 mark scheme points. So what is going to happen? Protons and electrons are released. It gives up its hydrogen, which breaks up into protons and electrons. Or you can say hydrogen is released or protons and electrons are released. So you could have said hydrogen. It's a slash. You could have said protons and electrons are released. Or you could have said reduced NAD is oxidized. So you could have said that. And for the second mark, where? At ETC. Or ETC is allowed. It's a standard abbreviation. Or if you said oxidative phosphorylation. So ATP is released, or the protons, sorry, are released. And where does it go? It goes to the electron transport chain where for oxidative phosphorylation. Technically a difficult question. B. The pH of the blood of an athlete decreases during a race and returns to its normal level after the race. The decrease in the pH of the level is caused by the presence of waste products that have been excreted by cells during respiration. Name the waste products. Name that are excreted and describe what occurs to these products to help return the pH of blood back to normal level. Now, unless you know this, you can do it. Otherwise, you can't be doing this question. This is a very technically. So the first point is lactate produced. Lactate is also called lactic acid. Lactate produced. Lactate taken to the liver. So where was the lactate produced? The lactate was produced in the muscles. It's taken to the liver. Here it is converted back to pyruvate. Converted to pyruvate. It is converted back to pyruvate. Then the pyruvate is converted to glucose. It is converted to glucose or maybe even stored as glycogen if it's not needed. Because you see, it might not undergo the different steps because if we have enough ATP, nothing is going to be happening. Carbon dioxide is produced. So when the lactate is converted to pyruvate, now why is carbon dioxide? Because here, what are we talking about? Describe what occurs to these products to help return the pH of the blood back to normal level. When a person is exercising, body is aerobically respiring, a lot of carbon dioxide is being produced. Now, carbon dioxide goes into form bicarbonate ions and all that. So there are carbon dioxide or there are pH receptors. And these pH receptors are going to detect this. Carbon dioxide goes into alveoli. So from the muscles, it's going to reach the alveoli. And there is going to be an increased breathing rate. That is when the person is going to breathe deeply and more frequently. So increased breathing rate. And hemoglobin acts as a buffer. Hemoglobin acts as a buffer, which helps in the pH not changing too much. Acts as a buffer for carbon dioxide. Next is question 3. In respiration, most ATP is synthesized during oxidative phosphorylation. Some ATP is made by substrate-linked reactions in glycolysis and Krebs cycle. Describe how ATP is made by substrate-level phosphorylation. Describe. So basically what happens in substrate is inorganic phosphate. Inorganic phosphate added to ADP. ADP. Or you could have said ADP plus PI. PI is inorganic phosphate. And you could have given a named phosphorylated compound like ADP is converted to ATP. So named phosphorylated compound. You should have referred to that. Like in glycolysis, it's phosphoenol pyruvate, PEP, which is converted to pyruvate. So the phosphate is given up and the phosphate is added to ADP and we say this is substrate level phosphorylation. So PEP is converted to pyruvate. Then we come on to the B part of the question. It says lipids can be metabolized to provide ATP. The enzyme lipase hydrolyzes lipids to glycerol and fatty acids. The hydrocarbon chain of the fatty acid breaks down the smaller 2C compounds. Each 2C compound reacts with coenzyme A to form acetyl-CoA. Name the covalent bond. That's a very simple one. This is a direct question. Ester bond. So the ester would get you another mark. State the role of acetyl-CoA in respiration. It is a carrier of the 2-carbon acetate or the acetyl group. It is a carrier of the 2-carbon acetyl group to the Krebs cycle, or you can say oxaloacetate. So it is taking the 2-carbon acetyl group or the acetate to the Krebs cycle to combine with oxaloacetate. Then part 3, explain why lipids have a higher energy value than carbohydrates. Higher energy value than carbohydrates. Why do lipids have a higher energy? It's a very simple thing. We keep on saying this again and again. When I teach you, I also talk about this. More CH bonds, or you can say more hydrogen. More CH bonds or more hydrogen, so produces more what? Produces more reduced NAD or NADH. Produces more reduced NAD. Produces more reduced NAD. More oxidative phosphorylation. More oxidative phosphorylation. Or you could have said more chemiosmosis or more aerobic respiration. So more oxidative phosphorylation, and you know every NADH gives you 3 ATP, so produces more ATP. So it produces more ATP per unit mass, or you could have said per gram. The C part of the question says the respiratory quotient RQ is used to show which substrate is being metabolized by cells. It can be used by the equation below, molecules of CO2 or molecules of oxygen. Lauric acid is a saturated fatty acid found in coconuts and has a chain of 12 carbon atoms. Complete the equation below. So you have to balance the equation C12H24O2 plus, no, this will become 17 because we have 12 here and 12 here, 24. So we complete that. We have 12 and 2, 14 here. So 17 left. So 17O2. Calculate the RQ value. RQ value will be CO2 is 12 and this is O2, so 12 over 17, and this is equal to 0.71, and that should have got you the 2 marks. And question 4, figure 6.1 shows transmission electron micrograph section through the mitochondria. Table 6.1 shows some structures and compounds involved in aerobic respiration. Use the letter P or the letter Q from 6.1. To complete table 6.1 to show the location of the structures and where the compounds are used. So ATP synthase, what is its location? P, inner membrane. If you look at it, what is P? P is the inner membrane and Q is the matrix. Acetyl-CoA. Acetyl-CoA takes place in the link reaction. Link reaction takes place in the matrix, so that's Q. Phospholipid bilayer is P, the inner membrane, the outer membrane. These are all phospholipid bilayers. Then oxaloacetate would be Q because that is in the Krebs cycle, and Krebs cycle takes place in the matrix. Now, looking at the B part of the question, the number of mitochondria differ from cell to cell. Cells also vary in size and volume. Table 6.2 shows the mean number of mitochondria per cell, the mean cell volume, and the mean number of mitochondria per cubic meter of 3 types of mammalian cells. So fat cell, heart cell, and liver cell. So mean number of mitochondria is given, mean cell volume is given. Calculate the mean number of mitochondria for the liver cell. So this was not done. You had to calculate that. Please remember you have to give it in the same form, 10 raised to the power minus 3. That was the important thing. The maths was very easy, but you had to give it in the same way. When in a table you have to fill in a blank, you must give it in the same units, in the same number of decimal places. So 16.00 into 10 raised to the power minus 3. Write your answer in table 6.2. That is what you have to do. So you have to write this here in this column here. Suggest reasons for the difference in mean number of mitochondria per cubic meter between the fat cell and the heart cell. So in mitochondria, respiration produces ATP. In mitochondria, respiration produces ATP, and you all write produce energy. No, energy is released. So mitochondria, you cannot be writing that what you wrote in O levels. So in mitochondria, respiration produces ATP, heart muscle contracts. The word contracts has been underlined. If it has been underlined, it means it's an essential mark scheme. It's a word which you must use. That's another point. And more active needs more ATPs. If there's more muscular activity in a certain organ, then that will require more ATP. Then it says, suggest why it is difficult to compare the ability of the three types of cells to respire aerobically based only on the mean number of mitochondria per cell. Yeah, because mitochondria vary in size, might have 5 big mitochondria or 10 small ones. And also the surface area of the inner membrane is variable. So these are the things. I always say suggest is very difficult. It's only for the students which actually do understand a high level of biology, and the suggest is always difficult, and I often ask you to just leave it because don't spend hours on it and not even get that 1 mark. Coming to the last question, which is question 5, figure 6.1 is an outline diagram, shows 2 stages of aerobic respiration. So you see pyruvate to acetyl-CoA, this is a link reaction, and then we have got the Krebs cycle, and you can see step 1 citrate is formed, and then citrate. Step 2 Q becomes reduced Q, so there's reduced NADH, and then we have again Q, and then we have reduced R, and then we have reduced Q. So if you know that it's NAD and FAD. With reference to figure 6.1, name stage X. X is the link reaction. State the precise location of stage Y, matrix of mitochondria. State the numbered steps in stage Y in which decarboxylation occurs. So decarboxylation occurs in 2 and 3. State the type of reaction by which ATP is made during step 3, that's substrate linked phosphorylation, or you could have said also substrate level phosphorylation. Both are allowed. Name substances Q and R. Q was NAD and R was FAD. You can see that why? Because R is got to be produced 3 times. So sorry, Q is produced, so Q here, and Q here. So 3 NAD, and R had to be FAD because FAD is produced 1 and NAD is 3 in 1 Krebs cycle. So this is how you were going to figure this out. If you knew this very well, then you could figure it out. The last part is uncontrolled cell division can lead to cancerous tumor. Many cancer cells break down the amino acid glutamine and convert it to 5-carbon intermediate compound, which is shown in figure 6.1. Suggest how the breakdown of glutamine can lead to the production of ATP in a cancer cell rather than directly produced during step 3. Now, reduced NAD or FAD will go to the ETC where oxidative phosphorylation or chemiosmosis will take place. So glutamine 5-carbon intermediate, which is shown in figure 6.1. So this was what you had to see, that this was shown in figure 6.1. Now if you look at this diagram, this is the intermediate compound 5-carbon. And so after this glutamine could then be resulting in the formation of Q and R. So that means NAD and FAD. So Q would still be used here. Sorry, Q would still be forming reduced NAD and reduced FAD. So look at the question. Suggest how the breakdown of glutamine can lead to the production of ATP in a cancer cell other than the directly produced during step 3. So glutamine, then figure 6.1, this is what you had to figure it out. Okay, thank you very much. Thank you, and that is a few questions of respiration, and I will try to do the next chapter after this.